Here at the Oakley Lab at the University of California, Santa Barbara, we investigate the origins and evolution of complex traits. One complex trait that we study is bioluminescence, the production of light by a living organism. Animals produce light by using proteins called luciferases to oxidize or add oxygen to substrates called luciferins. Bioluminescence systems are incredibly diverse and independently or convergently evolved over 90 times across the tree of life. Luminous organisms include dinoflagellates, shrimp, fungi, and annelid worms. Because these organisms convergently evolve bioluminescence, they use different luciferases and luciferins. At the Oakley Lab, we study crustaceans called ostracods, commonly known as sea fireflies. These small crustaceans are about the size of a sesame seed, but produce large clouds of bioluminescent mucus. Luminous ostracods have a light-producing organ called the upper lip, which produces bioluminescence to deter predation. Some species of ostracods in the Caribbean use their bioluminescence for courtship, much like how terrestrial fireflies flash their lanterns to find mates. Here we have some dried ostracods in a 2-inch tube. If we add water and crush them, this process releases luciferase and luciferin, which reacts with oxygen to produce bioluminescence. We observe that the light intensity is decreasing over time. This is because luciferin is getting oxidized, and once it is oxidized, it cannot be used for light production. On the other hand, luciferase does not get destroyed in this process. We have just seen that ostracods use luciferase and luciferin to produce light. The gene encoding for the luciferase protein has been identified, but we do not know how ostracods biologically produce or biosynthesize their luciferin. To tackle this question, we will use thin layer chromatography. But first, we need to extract luciferin from the ostracods. Here, we have some dried ostracods with methanol in a tube. If we crush them in methanol instead of water, no light is produced because methanol destroys or degrades proteins such as luciferase. This process allows us to extract luciferin since the degraded luciferase cannot react with it. Next, we take the tubes containing crushed ostracods in water and crushed ostracods in methanol and use a centrifuge to spin them. This process separates the water or methanol from the cellular debris. If we add the luciferin extract to the luciferase extract, bioluminescence is produced. This means that we successfully extracted luciferase and luciferin using our protocols. Once we confirm that we extracted the luciferin, we will spot the luciferin extract on a TLC plate. The stationary phase of this TLC plate is silica gel. We spot the plate several times, waiting for the spot to dry in between each time, so that we will have enough sample on the plate. We can confirm that our sample is spotted correctly on the TLC plate by using UV light. Because luciferin is fluorescent, it is visible under UV light. Now we place the stationary phase into a beaker with our solvent, the mobile phase. Our mobile phase contains methanol and chloroform. As the mobile phase travels up the stationary phase, the small molecules in the luciferin extract will migrate. How much the small molecules migrate depends on their polarity. Once the mobile phase nears the top of the stationary phase, we will remove it from the beaker and quickly mark the solvent front, which is the distance that the mobile phase traveled. After we run thin layer chromatography, we can use the UV light to visualize the luciferin and any other fluorescent molecules present in the luciferin extract. The luciferin spot here is faint, which suggests that we need to spot the stationary phase with more luciferin extract for our future thin layer chromatography runs. In the lab, we use thin layer chromatography along with DNA sequencing techniques to explore how ostracods produce luciferin. This work will allow us to discover how bioluminescence evolved and how this tiny oshakot can produce such brilliant light.